Afternoon, everyone. Welcome back from lunch. I know some folks are still trickling in from some of our breakouts. Our next session is called Dystrophin, the missing muscle protein. So the focus of this session is we're going to start with a talk by Dr. Stan Nelson, going through a little bit of helping understand what we're talking about when we discuss dystrophin expression. We obviously talked a little bit about that during the gene therapy sessions yesterday. You're going to hear more about it during this session, so just a little bit of a level set for everyone around kind of how we think about this. Then we'll go through some of the investigational products that are being developed that are looking at restoring dystrophin. And then finally, we'll kind of take a look at some more maybe novel approaches that are still kind of sometimes in the lab or very early stage kind of development, just to take a look at maybe where some of the other approaches might be headed. So to start off, we're gonna have Dr. Stan Nelson from UCLA join us on stage to kind of give us an overview on a better understanding of dystrophin expression and how we look at this in clinical trials.
Thanks, Eric. So I'm going to jump right in. And so I put together a talk to sort of get us oriented around dystrophin abundance, and what we do know and what we don't know, and some of the difficulties actually of quantifying dystrophin. So I'm going to start with this slide, and a lot of this is review information, and it's review information for me, it's review information for Kevin, and we're all thinking about this all the time. So it's not like we don't keep going back to the basics ourselves. But dystrophin is that missing muscle protein that's encoded by the DMD gene. It's on the X chromosome, which is why it's X-linked recessive, of course. And the dystrophin itself, for the functional version, is a long linear protein that is, as indicated, I'm going to be pointing way over here, I guess. So this long linear protein has a portion here that's binding inside the muscle, and this portion here is binding at the surface of the muscle. And the best explanation that's the simplest to understand is that it's acting a bit like a molecular shock absorber, protecting the muscle membrane from damage as forces are trying to be transmitted as we're all contracting our muscles. And it's missing, and that leads to periodic muscle damage over time, so particularly eccentric contraction-induced injury. And every single muscle fiber in every single muscle bundle, making up an entire muscle, has dystrophin on it all throughout your body. And then as we look at different muscles progressing, this is an image from imaging DMD, some muscles are progressing more slowly, and some muscles are progressing more quickly with Duchenne. And that's why we see the characteristic pattern of a presentation in the center of the body, and the muscles of the glutes and your hamstrings and your quads are affected earlier, and then muscles of the lower part of the legs later, shoulders a bit later, etc. So there's a characteristic clinical pattern, and that's reflected in which muscles are being damaged. But all the muscles actually have about the same amount of dystrophin. So even within this one short little snippet, we know that dystrophin it's the entirety of why the disease occurs, but it's not the sole answer, but it's a really critical answer, and so we now have ways to quantify it rather well. This is a boy who actually has some dystrophin in his muscle, and these are parts of his muscle fibers that don't have any dystrophin, and different parts of his muscle fibers that actually do have dystrophin. The dystrophin is indicated as this darker brown stain surrounding each individual myofiber. And that's what we're trying to quantify when we're looking and asking the question, how much dystrophin does somebody have? That's naturally occurring dystrophin. So virtually everybody with a naturally occurring dystrophin who has Duchenne or Becker muscular dystrophy has a single mutation, but that single mutation can be complex. It can be a large deletion, that's most common, deleting 1 exon, 2 exons, 3 exons, up to dozens of exons, up to the whole gene actually has been observed. So those are mutations across the full-length gene up above, where parts of it are missing. So for the most part, we're looking at just absence of dystrophin in most Duchenne individuals. And now we have a variety of other types of dystrophins. These other dystrophins, we're also calling them dystrophins. They're microdystrophins, mini-dystrophins, but they're made synthetically, essentially, in the lab, so that they have a different RNA sequence, actually a very different RNA sequence, very different DNA sequence, but they largely made the same protein sequence. That's what the companies have all been doing as they make each one of these products in the laboratory, so that they can ultimately make a very highly effective, that is, a lot of the protein is made in individual boys' muscles is the goal. But it's not exactly the same as the naturally occurring dystrophin, and then it's also broken up in ways that are never naturally occurring DNA mutations. So, but we want to now quantify these other types of artificial dystrophins as well in boys' muscles. So part of the reason I draw this slide up here is to orient you to part of the way that we go about trying to find a protein or quantify a protein is often with antibodies. So these little boxes here are indicating different antibodies. And this antibody is designed, was built, was found by trying to find an antigen, a sequence in the protein. The antigen's now the thing the antibody's going to react against. The antigen is dystrophin, but it's not all of dystrophin. It's actually just a tiny little part of dystrophin. So we often love to have antibodies that this one here only binds to this region. It doesn't bind to the rest of the dystrophin. The next one over binds to this region. It doesn't bind to the beginning or the whole rest. This one here binds to the middle. It again doesn't bind to the beginning or the end. This one over here only binds to the end. It doesn't bind to the rest. So when we talk and try to quantify dystrophin, as we're thinking about this, and we're critically looking at each other's papers and not publishing them or publishing them, depending on how it goes, we're very critical of what these things mean and how they work, essentially, and what the information is. But each antibody can only be reporting on a part of the protein. But if you had a mutation down here at the beginning of the gene that was leading to some dystrophin production, and this was positive, it's fairly likely that the whole protein is positive. So these are 3 different boys' muscle biopsies from a relatively slightly mildly affected Duchenne with the small patches of dystrophin restoration. Some of the text on these have been wildly changed. I can't even read the fonts up there. But this boy has an early mutation, exon 3 to 7 deletion, and this young man has a 50-51 deletion. So these are going in order of relatively lower dystrophin, intermediate dystrophin, and higher dystrophin, consistent with how their clinical presentation was. So that makes us feel good about what this dystrophin means. So one way that we look for dystrophin, one common thing that you'll get information about is immunofluorescence. So this is taking the antibody that's going to bind to a specific region of the protein, labeling it in some fashion with a fluorophore, with a fluorescent molecule, and then hybridizing it often to a muscle section caught in cross section. So the muscle fibers now look like circles, like these little rounded areas here, and each one is going to be decorated with fluorophore, indicating that the antibody bound. And you can quantify that. Actually, Kevin has done some of the most beautiful work in this, trying to very carefully find what where the membrane is in every muscle fiber, and then quantifying how much fluorescence intensity is there so that one could report out the quantitative amount of dystrophin at every myofiber. So then this would be giving us different information depending on which antibody we're using. If we're using that first antibody that's binding at the N-terminal, it could actually give us a dramatically different answer than the second antibody, because that part of the protein may be missing, not necessarily that the whole dystrophin isn't present. And then we can typically take a couple of bits of information from this as well. So if some of the fibers are positive and other fibers are negative, that will come up with a different measurement from the same type of data for percent positive dystrophin fibers, or as an alternate statement of the alternate way that they would be commented on is what percentage of the muscle fibers are positive. So to do that, you're also taking cross sections of muscles, looking at them carefully, using some sort of stain to identify where all the muscle fibers are, finding that region of the slide, essentially where the membrane is, and therefore where the membrane is where dystrophin could be expressed, and then quantifying if that fiber had expression or that fiber didn't have expression. And this is how complex those images look, and trying to do that across the whole slide is actually quite challenging to do. But this has been done, and you can set various thresholds as you move through the muscle fibers to be able to quantify both immunofluorescence-based dystrophin intensity and the percent positive fibers. So I threw this slide in just to highlight how challenging this can be. So we sort of think of muscle as being a bit of a monolith, or there's a typical muscle, but there's wild variation in muscle. So all of these slides here are muscles taken at the same intensity, showing dystrophin staining, but also showing that the myofiber size can sometimes be quite small, can sometimes be quite large. And a pathologic feature of a Duchenne boy is that he has both unusually small fibers and unusually large fibers. So exactly how you go about quantifying the surface intensity of each one of those large and small fibers actually is quite challenging. So then there's a Western blot. So many of you will have gotten some sort of quantitative assessment. All of the companies that are working in this space are generally using something that's like a Western blot. And this is a visual representation of this. And this actually has all of its own individual wrinkles and issues with it. So to do this is named a Western blot in honor of the person who did this with DNA, which is Ed Southern. So it's a Southern blot. And because this isn't DNA, it's the RNA is then a Northern blot to be not DNA. And then a Western blot is because it's not North and South, it's West. So this is a Western blot. So take with that what you will about what scientists think about and how they name things. So Western blots are just the term that we all use for when we're trying to probe something that's like a Southern blot, but we're going to be looking for a protein interaction. So now instead of using a DNA probe or an RNA probe for a Southern blot or a Northern blot, this is now a Western blot using an antibody probe virtually all the time. And then in this, you run the proteins out on a gel, so using electricity to put electrical force on them through some sort of polyacrylamide to give resistance to the molecules. Big molecules go through the gel much slower, small molecules go through much faster, and you can accurately size proteins using this sort of technology. And then once they're spread out, these are then transferred onto a membrane, and then that membrane is probed with antibodies that typically have either fluorescence or radioactive in a previous generation, or other quantitative means to assess how much protein is present. And then you quantify that. So you typically have a sizing standard to know what size protein you're looking at, and then a quantitation to see indeed how much protein there is. And this is actually a good, reliable measure, and indeed with titrations, you can get a pretty good sense of how much protein is abundant. And this has been, of course, published in hundreds of thousands of papers. But with all methods, it has lots of issues. So dystrophin is actually a little hard to extract because it is membrane bound. Dystrophin is a really big protein. A really big protein is actually hard to transfer from the gel onto a membrane. If dystrophin happens to be smaller, it actually transfers easily. So detecting a smaller protein is much easier than detecting a larger protein. And all of these would indeed factor into how well we quantify. So people spend a lot of time and energy putting essentially calibration standards onto these sorts of plots. And that's part of the reason why the data is not as tightly robust as we would always like, particularly when we're at the lower end of the dystrophin expression region, which is where we like to be. We'd like to move from no dystrophin to some dystrophin and reliably say that it's present. So that has been done in clinical trials. It has been done to demonstrate some dystrophin induction in pretty much all of the drugs that we have an accelerated approval for and will continue to be done, but there's methods improvements that could be benefited here. So at the bottom is a sort of an example of these Western blots showing virtually no dystrophin here. The gel is actually being exposed much more than this one up here, which is a set of Becker proteins, which are often a little smaller, running a bit smaller than the wild-type dystrophin. And then these are wild-type or reference typical dystrophin running up here, about 427,000 Daltons. And then this is a quantification from these authors here of typical Duchenne distribution of quantifications, individuals that were clinically identified as Becker and then healthy, showing a fairly substantial range of quantifications from some of these techniques. And this is actually just highlighting that you're using one method and a different method, a mass spec here and Western blot here. You don't always get the same answer, but in general, the methods are giving fairly similar answers. So, there's various different sensitivities for what mass spec, different sensitivity for Western blot, various different sensitivities depending on how we do the Western blot and what antibody we use. So this is an approach that is using capillaries, so trying to replace the gel electrophoresis by doing the electrophoresis within capillaries so that issues of where the protein is moved in the gel, which is shown up here, can be addressed by just measuring protein within individual capillaries. And this has some huge advantages, being able to load about 1/20th to 1/100th as much protein and still get nice quantitative data in terms of how abundant a given protein is, and being able to look for lower and lower levels. So these are some technical improvements that are being brought into the field over time. These are actually all low-level dystrophin abundances, showing some variability in terms of when our children are diagnosed with Duchenne muscular dystrophy and we have an out-of-frame mutation, the genetics tells us there should be no dystrophin. Occasionally, that's not quite true. So some of those individuals actually have low-level, few percent dystrophin, and indeed can be measured by a few different methods and quite reliably detected. And then I throw this one in from that same paper that I was just showing you, that this is the average of what's happening. All the reds are dystrophin abundance quantified through some of these careful methods. So there's a variation of what's been observed in healthy individuals. These are blue are individuals who are clinically diagnosed as Becker muscular dystrophy, and down here in orange are very low levels of dystrophin detected through this method in those individuals diagnosed with Duchenne muscular dystrophy. Showing a very big difference. No question this is the cause of the disease, no question no dystrophin is the issue, but also highlighting that there can be substantial variation even in healthy individuals. So, the commentary here is that we are all cautious about how we go about quantifying dystrophin. There are not particularly great clinical methods to do dystrophin quantification that we can rely on as clinicians and send these off and order them. So it's actually quite a difficult thing for you to ask of your physicians at this time, but it's something that's critically important in virtually all of the dystrophin restoration strategies, and probably something that's going to come back into the field as many of our children are dosed and treated. We will have these questions about how much dystrophin is there, how reliable is this, how could we compare. The other comment that I might make is that it's actually quite difficult to compare study 1 from study 2 from study 3, partly because of the differences in methods, where you're setting the gain, sort of how carefully you're looking for the dystrophin, whether you're looking for 0 to 0.5 or 1% versus you're looking for moving from 1% to 10%. So it's quite difficult for us to compare the information from one paper to another at this point, and that should be worked on over time. So I would and this has been commented on for quite a long time, but a universal comparator that can be embedded in virtually every dystrophin quantification experiment is actually a critical ingredient that would help us move forward to give better information about really how much dystrophin is in individual muscles along the way. So with that, I'm going to step aside and we'll move on to our next speaker.
Thank you for that, Stan. I think that'll help as well as we move into our next talk by Dr. Kevin Flanigan from Nationwide Children's Hospital, who is going to walk through a number of the investigational products that are currently in development looking at exon skipping. So that, I think, talk of a little bit of understanding dystrophin expression will help as we kind of walk through, and you're going to see a lot of that kind of data presented in this talk.
Thank you very much, and thanks, Stan. That's a great introduction, great talk. Really leads well, I think, into this one. I'm presenting here really on behalf. The structure this year was to allow each of these companies to provide slides that we could then sort of edit a little bit and present their data. So some of them I'm more familiar with than others. Oh, this is not the uploaded one. Sorry. John, do we have a chance? We had an uploaded version that was named Final. Is it easy to quickly switch to that one, do you think? We just had a couple of extra slides, sort of a background for this. If we can't do it quickly, I don't know, maybe I can press on ahead, or I can describe it by interpretive dance, or... Okay, they'll be able to do it, sorry. All right, so this guy walks into a bar. I got nothing up here. No, I'll vamp for a minute to say I do want to make sure we include a little bit of description about exon skipping, and I think the critical thing, I'll just give you a highlight of what the introductory slides was about, is really this concept that Stan pointed out, the difficulty in comparing among different studies just to quantify dystrophin. That's critical. These methods, these assays are different. But also, the important thing is, when we're talking about what we're quantifying, it's not just how much there is, but how functional that is. And how can we gain experience from information from patients, from large numbers of patients, to try to understand what the functionality of a given protein is. And I'll show you a couple of examples of that in the introductory portion, I think. That's all I had for vamping, actually. So I think we're on it back there. So I'm going to talk in general. I'll show you some of these images in a minute to help with it. The basic structure is this to keep in mind. When a gene is transcribed from the chromosome, it comes off as a single-stranded, what's called pre-messenger RNA. And that single strand contains both the exons and the introns. Those are, so that molecule, soon after its transcription, a set of molecular machinery, proteins called the spliceosome, attaches to that pre-messenger RNA and literally translocates down the pre-messenger RNA molecule to where, and the spliceosome recognizes individual sites where splicing is to occur. So there are sequence motifs that say, hey, I'm an exon, splice me here. That gets put together into what we call a mature messenger RNA. And there's a couple of critical features that are important about this in exon skipping. All of the talks today are about exon skipping. The critical feature is that you have to have a single-stranded RNA to do that. So if you take a fragment of something that binds to that messenger RNA, the pre-messenger RNA, an antisense oligonucleotide, something that binds in the opposite direction, pairs in the right way, and hides that single-stranded exon definition site by that little now double-stranded fragment, the spliceosome can't do its job. So fundamentally, that's what exon skipping is. And when we talk about antisense oligonucleotides, we can make many different versions of these, and some of the companies use different chemistries to do this. The chemistries include different chemical backbones for stability of the antisense approach. One of those is called the phosphorodiamidate morpholino oligomers. So I'll refer to those in the talk as PMOs, just not to say that word again and again. So antisense oligomers are what the effector molecule is for exon skipping. Exon skipping therapies, as we'll mention, there are 4 commercially available exon skipping therapies right now, directed at exons 45 and 51 and 53. Those include eteplirsen, golodirsen, casimersen, and viltolarsen. Some of you might be on that, or your children might be on those. And all of what we're going to show you are improvements, attempts to improve the efficiency of the exon skipping from those really kind of naked PMOs, as we refer to them. So the different approaches that we'll touch on momentarily include first conjugating them to some sort of molecule that can help those improve penetration into the cell, and that's a key thing. Our slides are up, so I'll continue from there for hold that thought. And I do apologize for that, but those general concepts, I think, will show up in just a minute. I'll begin by just saying financial disclosure. I had forgot to do this in my talk yesterday. I'm going to talk on a couple of things today that I'm involved with studies on, but I have no other financial kind of interests. This is just a reminder of the dystrophin gene and protein. At the bottom is a sort of a cartoon of how the protein binds at one end to the cell skeleton and at the other end to the cell membrane, anchored by dystroglycan. And in between, we have this region of repeat elements, and long ago it was determined that you could get rid of fairly large portions of this if you maintained an open reading frame and have a functional protein. And a reminder here that when we talk about the dystrophinopathies, there's a whole range of things that are associated with dystrophinopathy. This is a continuum of disease. I know that you all know this as parents and family members of people with muscular dystrophy. Sometimes people say to us, do I have Duchenne or do I have Becker? What's the amount of dystrophin that defines it? What exactly defines those things? This is really a continuum across the disease process. And really, the exon skipping therapies we were talking about today are generally, the idea is to move a boy from the more severe end of Duchenne into more functional Becker end. Now, what that means, it really influences how we'll interpret some of the results. Just a reminder, exon skipping therapies really depend on this idea of the reading frame rule. If you maintain a reading frame that allows you to make a partially functional protein, you'll likely have a milder disease like Becker muscular dystrophy. And if you ablate the reading frame and make no dystrophin, that's what goes along with Duchenne. But that's the whole basis of exon skipping therapies. But one reminder, just to emphasize this correlation, we have to note that all Becker microdystrophins are not the same. All these internally deleted things are not the same. Here I've marked 3 lanes with B. This is not our modern Western blot. Stan was showing Western blots. We have much cleaner ones now, but this is one we made years ago, I've shown for years. So these are all from patients with Becker muscular dystrophy. All of them are shortened versions of the protein. And the differences between them, one is 15% in lane number 2. Second and last lane is about 140% of the normal control on this blot. Yet the loss of ambulation age was 70 years in the man with 15% and mid-20s in the 140%. So it's not simply the amount, it's how functional that protein is. So I went over this while I was vamping there for a minute, what exon skipping therapies do. They mask an exon, it's spliced out. There are 4 such therapies that exist. And before I get into a discussion of the studies that are underway, I want to emphasize how we can use clinical information to predict what are best potential outcomes for that, with 2 examples. One is when we made a big effort with the help of the Parent Project Muscular Dystrophy Registry and other registries around the globe. We looked at all the patients who had what would be who had entries for patients who had any kind of dystrophinopathy, that was the equivalent of skipping exon 51 therapeutically. So let's imagine you could skip exon 51 entirely. Well, if we look at men born with that same mutation, what is the best outcome we could look for? And I find this very interesting. I still do. When we look at the description, we lump these as asymptomatic, meaning not even an elevation of CK, X-linked dilated cardiomyopathy, elevation of CK alone, clinical features of Becker muscular dystrophy, clinical features of Duchenne muscular dystrophy. And it's interesting to see that certain of these things, men born missing 1551, 40% of them are actually asymptomatic, and some are categorized as Becker muscular dystrophy. We see some, and 45 to 51, their only symptoms of about 40% of the people are elevations of CK without significant weakness. It suggests to us that these can be very highly functional versions of dystrophins that could be made by efficient exon skipping. We should keep that in mind when we make comparisons, for example, to microdystrophins, which may actually be very efficient, although that efficiency is shown in animal models, not from patients, not from real life. And I'll give one more example of this. This is a similar paper we did years ago to look at the equivalent skipping. In this example, we were pulling out patients who had exon 45 skip equivalents. And we noted that although we had many patients who had 45 to 47 deletions or 45 to 48 deletions, none of these all men, all of them, even of younger age, were categorized as Becker muscular dystrophy clinically. None of them lost ambulation before about 40. In contrast, the patients with 45, 46 deletions all were called clinical Duchenne at a young age, and 2 of them had lost ambulation before 12. I note this because in this exact same paper in the discussion section, we commented on the absence of 44, 45 deletions. They were not in our database at the time. They were not in the public databases at the time. And because the entry to those databases was via symptoms or elevated CK, that's the only way you would get testing back in the early 2000s. We postulated in that paper that these were truly asymptomatic mutations. And I want to hold that thought because some of the data I'm going to show you is from exon 44 skipping therapy, some of which shows great promise for restoring functional proteins. So it's possible that some of the skip equivalents are truly asymptomatic, the equivalent of normal dystrophin function. So with that in mind, I'm going to go into some of the things provided by the companies. I'll start with Pepgen. Pepgen's approach was to use a cell-penetrating peptide that conjugated to the active PMO, or antisense oligonucleotide, in order to get through the cell membrane. And I won't spend a lot of time on this. The Pepgen company has had really sort of, although the safety profile was fine, there was a mean maximal dystrophin increase of 0.59%, which was really not considered sufficient to carry on. So the company has voluntarily discontinued the development. They did ask us to make sure to share that they wanted to express their gratitude to the patients who took part in this study and to those of the community who helped them develop this. But I'm not going to spend much more time on the cell-penetrating peptide approach. Another approach is, there's 2 companies I'll talk about here, Dyne Therapeutics and Avidity, that use similar approaches. This is Dyne's sort of disclaimer language, but I want to concentrate here on the mechanism in this case. So the current antisense therapies that are approved, the naked antisense PMOs, rely on diffusion across the cellular membrane. Cell-penetrating peptides, I mentioned, it was an effort to sort of pierce the membrane to get across it. This is a different approach where there's a receptor abundant on all of our cells called the transferrin 1 receptor that brings iron into cells. It's highly enriched in skeletal muscle and in heart. So what Dyne and Avidity does it a little differently, but what they do is they conjugate the active PMO, the active antisense oligonucleotide, they conjugate it to a fragment, an antibody fragment called the FAB fragment. That FAB fragment, it then binds to the transferrin receptor, and through the natural uptake activity of that receptor, those PMOs are now brought into the cell. And they're brought in, and once in the cell, the conjugation link is broken. They make their way to the nucleus where they do their their exon, where they have their impact on exon skipping. So this is a clever approach. I'll just add now the only difference between Dyne and Avidity really, or the major difference between them is Avidity does it with a IgG, a full IgG molecule, whereas Dyne does it with this FAB fragment. Dyne has a trial, the Dyne 251, the DELIVER trial for patients that was involved dose escalation for patients. I'll note here that it enrolled patients 4 to 16 years old, looking at safety and tolerability, pharmacokinetics, and this dose escalation led to really the determination of a registrational dose of 20 milligrams per kilogram to be given every 4 weeks. This contrasts with the current crop of medications that are approved that are given weekly. So every 4 weeks is actually much less burden on the patient population to do it. So that registrational trial, I'll point out that the safety profile is really quite good. There have been patient with at the higher dose with a potentially related serious transverse treatment emergent adverse event of kidney injury and thrombocytopenia and pancytopenia, which resolved, and that patient has continued on the trial at the higher dose, and now the dose is 20 milligrams. It continues, and I'm going to show you some data from biopsy data at 6 months in these patients. Stan mentioned ways of measuring dystrophin, so this led to an increase to a raw data level of about 3.7% of normal levels of dystrophin. And there's many studies, as many studies do, an effort to normalize for the amount of contractile muscle by factoring the degree of fat and fibrosis across a muscle section. And using that, this is about 8.7% at levels of dystrophin, which again, observations, these kind of levels, I'll just sort of editorially add from studies like in my lab and Stan's lab and others, these are ones that we do expect to have significant influence on the progression of disease. Consistent with that idea, this is a fairly complicated slide, and I'm not going to dwell on too long in the interest of time, but I'll point out here a couple of different things. The blue bars are the patients who initiated and remained on the 20 mg per kg dose. The green symbols are those who started at a lower dose and then were moved to 20 mg per kg, and then there's ones who were on placebo alone. But you'll see there's evidence for clinical improvement in North Star Ambulatory Assessment, time to rise for the floor, 10-meter walk run, and this stride velocity, 95th percentile test. For those of you who are unaware of it, it's a method by which a accelerometer is placed on the limbs of a child so you can actually see what they're doing out in the community, and a measure that's been accepted by the European Medicines Agency is the 95th percentile of the velocity a child takes. So it's one you'll hear more about in coming years. So the updates to this registrational expansion cohort is fully enrolled. We expect to see more data in late 2025. And Dyne has announced they anticipate submitting for accelerated approval in early 2026. We anticipate more information at the end of this year. Now, I've lumped next to Avidity Biosciences really because they use this similar technique, the transferrin receptor 1 mediated entry into the cell. I'm going to call their drug Delzota. It's easier to pronounce than the real name of it. So here we're targeting not exon 51, but exon 44, which I mentioned to you before. An outcome might be particularly good for patients who have deletions of exon 45. So again, it uses this in the TFR antibody to enhance delivery of the active drug into muscle. So the EXPLORE trial was designed to evaluate the safety and efficacy of this product. It's active and fully enrolled with boys between 7 and 27 years of age. And I'll give kudos to Avidity for including the older patient population in here. These boys had to have clinical diagnosis of DMD that was clear before the age of 6, and both, again, ambulatory and non-ambulatory patients. The primary endpoint is safety, really the incidence of these treatment-emergent adverse events through study completion. Secondary endpoints include change from baseline in serum CK at study weeks 24, 48, and 102. But a variety of other exploratory endpoints are included. And they didn't provide for me here sort of graphs of this, but there's an average increase of 25% of normal in dystrophin, an increase of 25% of normal levels in dystrophin production with both doses studies. And strikingly increased reductions in creatine kinase to normal or near normal in 80% of these patients, which might reflect the high exon 45 deletion is the most common single deletion that exists in patients with DMD, and it's enriched in the enrollment of this trial as well. So I suspect that that accounts for some of these great numbers. So a favorable safety and tolerability profile, and here again at a relatively low dose, 5 mg per kg, with 6-week dosing every 6 weeks, which is again an improvement in tolerability. It's been granted fast track status, and there's planning for a filing for accelerated approval at year end here at 2025. Those plans include then plan for a phase 3 confirmatory trial, and we await information on whether that will be done ex-US or how that'll be actually organized. But the program really will advance to additional exons, including now exon 45 in IND-enabling studies. We'll return to exon 51 with Biomarin's product. Biomarin has some chemical modifications to the backbone that improve stability and binding, and targets a different site than the currently available exon 51 therapy. It is again given as an intravenous infusion. So the study 201 is underway for boys who it'll be enrolling 18 boys who are 4 to 10 years old that have a mutation amenable to exon 51 skipping. Again, as most of these studies require stable dosing with steroids for 12 weeks before beginning the trial. There are other rules, of course, not having other exon skipping therapies and not having received gene therapies. If you choose to participate in this, it's going to involve infusions, standard tests, weekly clinic visits, and it will last 48 to 56 weeks. And if safety is acceptable, patients would continue in an extension study. Now, this is underway overseas. It's actively recruiting cohort 3 in the UK. I'll give you at the end the people to contact if you wish to enroll in it. 351, though, is going to go in next to a planned phase 2B study that will be 72 weeks long, involving patients from 36 participants. It will be in the US as well. And we'll take some patients with a weaker score than one of the measures used originally, the Brooks score. They postulate here that, one of the things about all exon therapies, in fact, we have to think about is, as dystrophin is translated, it takes a long time to reach a steady state of dystrophin expression. So they have planned biopsies later, as you'll show. The idea here is that this might increase slowly. So they're going to look if there's a difference between dystrophin expression at week 25 with a subsequent biopsy to see if there's continued increases in it as well. So I'll add here, you're welcome to contact Biomarin for medical information about which sites in the US will be doing this. So I'll talk next about Entrada, which uses yet a different way to get antisense oligonucleotides into the cell. In this case, Entrada is, I'm going to skip over some of this to say what the idea here is, because the targeting is inside the cell, they're using something called the endosomal escape vehicle, sort of an envelope, a way to package antisense oligos to improve uptake into muscle. The idea here is that this chemistry allows uptake within endosomes that are taken into cells, and then once in the cells, again, it's active uptake into myofibers. So this, the chemistry of it allows to have efficient escape from the endosome and travel then into the nucleus to do its work. There are 4 programs, a 5th in myotonic dystrophy, 4 in DMD that Entrada have underway. I'm going to talk about Entrada 44, their exon 44 one. The only data that exists right now so far is this from phase 1 study in healthy volunteers who are given, 32 adults were given a single dose of this, 6 milligrams per kilogram, and a dose escalation study. Outcome measures were measuring tolerability and target engagement as measured by exon skipping. So it was well tolerated. There's a headache as a common event, no adverse findings from this single dose of it, but there was evidence for target engagement. First off, there was high plasma concentrations of this, suggesting that it's at relatively low dose, it gets across the bloodstream, and favorable evidence for target engagement at this single dose in individual patients with increases of exon skipping to levels of above 40%, as you've seen in the right. So we've got a robust target engagement. So this is kind of rapidly expanding. The study initiation of relevant trials in Europe, in UK and Europe, are expected later this year. And in the US, a multiple ascending dose study in patients will be in the first half of 2026. And we expect more information from the company on that. And you can contact Entrada as well. So, I'll just steer you to entrada.com. NS Pharma has different chemistries, some special components of what they're after. I'm going to talk about 2 programs, their exon 50 program and their exon 44 program. So the difference here is the backbone. There's 2 things. There's a sort of what's called stereo pure a version of the chirality of the actual molecule itself is in one direction, and there's another change chemically to the backbone that might improve its stability. So this study is currently enrolling a multiple ascending dose study with plans for an open label treatment for patients with exon 50 amenable mutation in boys 4 to less than 15 years old. There are some requirements for function, ability to complete the time to stand from the floor in under 20 seconds, being able to complete the time to climb 4 stairs, and again, a minimal 3-month stable dose of glucocorticoids with no prior treatment of gene therapy. It's going to look at endpoints of safety and tolerability and the pharmacokinetics of the drug in the bloodstream of boys with Duchenne dystrophy. And then to see about the induction of dystrophin expression in skeletal muscle by biopsy, along with motor function measures. So it's actively enrolling, and so far no safety concerns have been identified to date. It's available, I'll refer you to the clinicaltrials.gov up above. There will be additional countries, ex-US, starting to plan for screening in the end of this year. NS Pharma also has a program directed to exon 44. You wouldn't know it from the title. There's no 44 in the title, but it's exon 44. So this is a study that's currently enrolling patients. Again, the design is what's called a multiple ascending dose study. And once a dose is then picked, an open label treatment for 24 weeks. Again, very similar criteria, boys 4 to less than 15 years old. I'm going to just skip to say that it's kind of identical in its safety as an endpoint, tolerability, evaluate dystrophin splicing, evaluate dystrophin expression. Now, last year at the MDA, Dr. Paula Clemens presented a poster that showed some of the preliminary data in an early phase 1-2 study in just 6 patients that did show a significant change of about 13% in the level of dystrophin from baseline to week 24, and clearly showed this was associated with exon skipping. So there's no drug-related SAEs and no patients discontinued. That led to this current next phase 2 clinical study. And again, there are additional, for exon 44, additional countries will be screening by Q1 2026. I'll refer you again to this clinicaltrials.gov site. And our representatives from Pharma are here too, if you're interested in talking to them. And I'll close with Wave Life Sciences. So Wave has yet another approach based on specific modifications to the backbone chemistry. So this is the Forward 53 trial. So it was shown, released in a patient of 11 patients that were treated with it, a 3.8% second improvement in the time to rise versus the natural history. That's the really outcome of the data that's important. Distribution to muscle fibers with high muscle tissue concentrations, and reductions in muscle fibrosis and CK, driven by decreases potentially in inflammation. I'll show you some pictures of that for a moment. So dystrophin expression really averaged at about 8% between 24 and 48 weeks, which is an improvement on some of the published or some of the approved therapies. I won't really belabor that. This is data that they've presented publicly suggesting some reversal of muscle damage across the majority of participants. An editorial comment here would be, we always have to be somewhat cautious in interpreting data like this. We really want to make sure we are looking at comparable regions of muscle, comparable muscles and so forth, but the data does suggest that there is some improvement in muscle pathology. And just to put that 3.8% second improvement in comparison, this is in comparison to the natural history cohort. So it did not stop the lengthening the time to rise, but it sort of slowed it down. So this is, again, something of a differentiated therapy. Again, it's potentially more tolerable in that it's monthly dosing rather than weekly dosing, and does potentially show benefit. So the Forward 53 trial is continuing. They intend to submit an NDA to support accelerated approval with monthly dosing, and there are additional clinical programs moving forward. So there's the contact information for Wave Life Sciences if you're interested in that. So I'm going to close by just a couple of reminders. This was a kind of rapid overview of multiple different programs. I think, again, editorially, I'll say it's quite promising. I've talked to many parents who have AAV antibody positivity that precludes them from Elevidys or one of the experimental microdystrophin therapies. Just want to assure you that that exon skipping therapies really are alive and well, and some of them look exceedingly promising. And I anticipate that in the coming years, we're going to hear more from one or more of these companies. And we'll keep this in mind as you have choices. Obviously, not all parents have the opportunity, not all amenable to skipping to each of these. But as a clinician and a scientist, I'm glad that these are moving forward with really second-generation exon skipping approaches. A reminder that in comparing any of these different therapies against each other or comparing them against microdystrophin, it's not only how much dystrophin there is, but how functional that protein is. And we learn how functional those proteins are from studies in patients. So thank you for all of you who participate in those studies. Approval of a microdystrophin vectors has been a landmark, obviously, but second-generation therapies are now in trials. And treatment considerations, as you think about it for his son, should take into account his own genotype, as we may be able to get to better therapeutic dystrophins by other means. So with that, I'll close. Thank you very much.
And then our final speaker for this session before we move to the open Q&A is Dr. John Lueck from the University of Rochester, who's going to talk through some novel approaches that are being looked at in the lab, or kind of really a little bit earlier stage, but kind of looking at some newer things to share with the community.
All right. Hello, everybody. I'm really happy to be here. It's the first one of these meetings I've been to, and it's been really great meeting all the scientists and, of course, all of the families. Before I start, I'd like to actually, I think we should all thank Eric. It's really hard to get scientists to get their talks in on time. It's like herding cats. So if we can give Eric a round of applause for getting these. All right, so we'll get to going. And I was trying to figure out where to start, so I thought the best place would be the beginning. So central dogma of genetics. So you have your gene, right, which is DNA, which is transcribed into pre-mRNA, which is then processed into RNA. That RNA, mRNA is then translated into protein. And so when you have a mutation in the gene, this results in an abnormal RNA, which then, of course, results in an abnormal protein that is harmful and results in a disease. And so there are many different types of mutations that you can get. You can get deletions, duplications, missense mutations, where you go from one amino acid to another. And then there's this other form which occurs in 10 to 15% of all inherited diseases, called nonsense mutations. And that's when you go from one amino acid to a stop codon, so where the process of translation is terminated. And so, and that's shown here. And so these diseases are generally more serious than ones with missense mutations, because you have complete loss of the protein. And for Duchenne muscular dystrophy, that's no different. And so what approach that we're taking in the lab, and several other people are taking, is to target the translation process. And so it's turning that stop signal into a go signal. And so what we're developing are what's called anti-codon engineered tRNAs. And I'll go into what those are here next. And so getting into the process of translation, what I've shown you there in that squiggly line is the mRNA, and the blue and purple ball is the ribosome. And so the ribosome binds to the mRNA and ratchets down 3 nucleotides at a time. Those 3 nucleotides are called codons, and those encode for 1 amino acid. So as it's moving through the mRNA, going through translation, you see that green cloud appear, that's the protein. And when the ribosome gets to the very end of the transcript, there's a stop codon. The ribosome is then disassociated from that mRNA, and you have a fully functional protein. When you have a nonsense mutation or a premature termination codon, you have a stop codon in the middle of the transcript, right? And so the ribosome, as it's translating 3 nucleotides at a time, will reach a premature termination codon. You have a truncated protein that's not functional. And another mechanism that occurs in this type of mutation is that the transcript actually is being degraded through this process called nonsense-mediated decay. So now taking a closer look into what's happening in that ribosome. And so you have this molecule called tRNA, and that's shown very artistically as that blue squiggly line at the top left. And every tRNA is encoded or is important for encoding a specific amino acid. So here I have represented that tRNA encoding a glycine, represented by GLY. And that amino acid is appended to the tRNA by this enzyme called aminoacyl tRNA synthetase, and it's a keyed interaction. The charged tRNA then gets brought to the ribosome, which is again shown by that blue and purple ball, by this protein called EF1 alpha. And EF1 alpha is I kind of think of it as the helmet for the charged tRNA. It keeps the amino acid from being taken off. And it's also is important for delivering it to the ribosome, where it participates in translation. And so where I have shown the blue squiggly tRNA in the ribosome, that is like a big speed dating situation there. So you have, I'm showing you this for glycine, but you have 20 amino acids, and you have several tRNAs that are coming in and sampling that sequence to see if it's right for them. And if it is right for them, they sit and they transfer that amino acid to the growing peptide chain, which comes off the back of the ribosome. And there are no tRNAs for stop codons. And so if this is why, when the ribosome gets to a stop codon, it dissociates and you have a truncated protein. Or if it gets to the end of the transcript, you have a full-length protein, but the ribosome does not continue through. So the approach we're taking is to take a naturally occurring human tRNA and editing the anticodon, which is the bit that interacts with the codon on the mRNA, to recognize one of the 3 stop codons. So there's a stop codon UGA, UAA, and UAG. That we call anticodon edited or ACE tRNA is still recognized by the enzyme that puts the amino acid on. It's recognized by that little helmet, brings it to the ribosome, and now when the ribosome gets to a stop codon, there's a tRNA present to then extend the peptide chain past that stop codon. And you see I have this yellow blob there. That's a protein called release factor. And the amount of tRNA present has to be higher than that of the release factor, and they're in competition with each other. Not like a true competition, but more like a horse race. So you need, whatever gets there first is gonna win. So if you have an ACE tRNA get there, you'll have extension, and you'll have a long protein. If the release factor gets there first, then you'll have termination of translation. So and the important thing, so I've shown you a lot of proteins and a lot of transcripts here. The only thing that you need to deliver is the ACE tRNA as a therapeutic. It actually uses the endogenous proteins and systems in the cell for it to work. And so therapeutically, that's really important because it's a very simple system. And so when we first started these studies, I didn't know anything about tRNAs. And so, I was actually, this was several years ago, I was thinking about, this would be a really easy thing to do. Why hasn't anyone ever done this? Turns out dozens, hundreds of people have actually done this before. I just didn't read the literature. And when I went to go read the literature, I realized why it was so difficult and why people haven't done a lot of different sampling of tRNAs. It's because there's 415 tRNA genes in the human genome, right? And they're all there for encoding 20 different amino acids. But if you look through all these other species, which I have listed there, you can see the number of tRNA genes are quite varied. But the most amazing one I find is the bush baby. It has 45,225 tRNA genes in the genome. And so the question is, why does that bush baby, which is shown there on the right, have 45,000 tRNAs? And I think the only answer I can come up with is that they're really important for expressing cute genes. And you can tell from the nice big eyes there. And so the question was, which tRNA were we going to pick to make them into these engineered tRNAs? And so I'm not going to go through this entire data set here, but we just chose all of them. So we screened 500 different sequences to find which ones worked the best for suppressing stop codons for all of the amino acids that would be necessary for rescuing the expression of a wild type protein. And so those are listed here, and you can see that those bars are basically how good they are at suppressing stop codons, how far they go to the right. And you can see that they're varied in their ability to suppress stop codons, and some are really good and some are really bad, but from here we can take the ones that are the best and move on. And so here I have listed the process of, the central dogma of genetics on the left, going from DNA to protein, and on the right I've shown you a table of codons. And so you have 4 nucleotides, and those are all mixed together in sets of 3 to give you encoding the 20 amino acids. And there are 19 of these codons that are 1 nucleotide away from a stop, and those are all the ones that will result in a disease-causing nonsense mutation. And so we have the ability to go in and make those all the wild-type protein with our library of ACE tRNAs, but if you think about that, you'd have to advance 18 ACE tRNAs to the clinic. And that would just not be feasible. So the approach that we're thinking about is to take one of them that we think will work the best in all of those positions and use that. And so we think that leucine is the winner, and we can get into that in the question and answer period if you want to. But one of the big reasons is that the leucine tRNA can be used to suppress all 3 of the stop codons while others cannot. And also leucine is kind of an innocuous amino acid when you think about proteins. It's not charged, it's not bulky, it's flexible. And so when you have mutations across where you have to maintain structure of a protein, it's rather innocuous. Okay. And so, and also what the benefit is that if you use leucine, you only have to advance 3 therapeutics to the clinic. And so I think tRNAs are like one of the coolest things. I didn't realize how cool they were when I started studying them, obviously. But one of the great things about them is that the entire expression cassette is about 155 base pairs. And so we heard yesterday from Jeff Chamberlain, and we've heard several times that dystrophin is an incredibly large gene that we'd have to deliver to get a wild type protein back in for gene therapy. Here, no matter what the mutation is, you always are delivering the same therapeutic, and that therapeutic would be a gene that is 155 to 200 base pairs in size. So that fits in AAVs and any of the therapeutic viruses that are available, but you can also deliver it as RNA. Right? So you can transcribe it, and that's the true form of it in its active state, and that's showing you that in the left bottoming the green squiggly line there. And people are working on this. There's a company, Entrada, that is encoding or that is delivering tRNAs as RNA using lipid nanoparticles. people are delivering suppressor tRNAs or ACE tRNAs as virus, which I've shown on the right, and you can put in multiple copies into one AAV because the cargo of an AAV is about 4.5, 4.7 kb. You can put in as many copies really as you would ever want. And then down the middle, I've shown you DNA, and so that's what we're working on in the lab. You can encode tRNAs from a small, what we call picovector. I won't be getting into that data, but we're pretty excited about it. So as a platform, there are over 700 nonsense mutations that exist that have been identified, and our goal is to have 3 ACE tRNA therapeutics for treatment of all or most of them. So what needs to be done? This is very early in the use of this technology, so we need to determine its safety. And the main concern with ACE tRNAs or suppressor tRNAs is that there are stop codons at the end of every reading frame of every gene, right? And so then there's the stop codon that causes the disease, which is early. And so you have to be reading through the disease stop codon, but not the natural termination codon that ends at every gene. And so that has yet to be, I think, determined in every tissue type, and especially muscle after delivery. And then we need to determine the best delivery mechanism. And as I mentioned, we can deliver these as AAV, naked DNA, and as RNA. And the next thing that we really need to worry about is delivery, and delivery, and delivery. And so I think delivery is going to be the biggest hurdle. Without being effective at getting them into skeletal muscle, we won't know if they're toxic, we won't know the benefit of them. And so we don't want to be putting the cart before the horse. So next I'll move on to another technology that is RNA-based, and it is U7 small nuclear RNA. And I'm just going to give you a brief overview on how it works. And so, as we've heard, dystrophin, mRNA is 75 or 79 exons. And here I've just shown a stop mutation in exon 42. And you can see from what I've shown you here on the diagram is that every exon is kind of keyed to each other, right? And so you have the pointy bit going into the recessed bit between exon 1 and 2. And so those need to be spliced together in order to stay in frame. And Kevin Flanigan discussed really nicely what is in frame and what is out of frame and how important that is. But you can also see that there are some exons in there that are square, right? And they're next to a square and they're next to another square. And you can splice those out or take them out and still stay in frame and have whatever that dystrophin would be minus that exon or several exons. And that's indicated here. So if you took out exon 42, for an example, it would stay in frame with between 41 and 42. And so this was covered a bit by Kevin. There's pre-mRNA, which includes this information called introns. Introns are spliced out, exons are put together, and that's where you get your mature or messenger RNA. And so what U7 sRNAs have been shown to do very effectively is you can bind them to specific sequences in the pre-mRNA and then force exclusion of that exon, right, by missing that splicing event, and then you'll have spliced out that exon. Sorry for the numbering there, that got a little wonky. That's a Mac to PC thing, I'm guessing. But you can see very clearly from the diagram that these exons are put together and they're in frame, but they're just missing that one exon. Okay, so what is the cargo for this approach? What is the therapeutic cargo? It is actually just an RNA. And so it's about 600 base pairs in size, including the promoter. And of course, that would fit very well on any one of the AAVs that is being used therapeutically. And so it makes this structure that has a sequence that's shown on the left with kind of looks like a comb, and that's important for binding the sequence of choice, and then a hairpin. It's only when you express that RNA in cells that it actually gets bound up by proteins that are present in the cell. And so you have your hybridizing bit that goes to the specific region of the pre-mRNA, and then that is bound by 7 proteins that form this kind of lariat around the stem. And those 7 proteins are important for stabilizing the RNA and actually delivering it back into the nucleus where it functions and have some function in coordinating the splicing events. And so importantly, if you go to the literature and you start reading about these, U7 siRNAs are a naturally occurring RNA, and basic research has discovered them as a therapeutic option. But they've been optimized, they've been changed or engineered to now bind different proteins to be an effective therapeutic. And so in the literature, they're sometimes called U7 SM opt RNA. And so here I'm showing you again the exons. And there's 2 mutations in exon 42, and I think this is a really good approach for U7 siRNAs. Those wouldn't be 2 mutations in one transcript, but let's say an individual with the first mutation and another individual with the second mutation. You would just then use the same U7 siRNA for both of those mutations. So in this way, it's a platform therapeutic. And so if you have a hot spot of mutations in any given exon, you would use just one therapeutic for all of them, and where you can remove exon 42, as I've shown you here, and stay in frame. You can also, because the therapeutic cargo is so small, you can fit several of these snRNAs into one AAV, and you can actually target several exons. And if these are mutations that exist, here I have shown exon 40, 41, 42, and 43, you would stay in frame and be able to remove all of them. Of course, it's important to stay in frame, and not every exon is amenable to this approach, and that's shown here in exon 43. If you remove it now, you're going to be out of frame, which then causes a premature termination codon downstream, and you'll get a truncated protein. So it's not a one-size-fits-all approach, but it's a one-size-fits-most approach, I would say. And recently there's been evidence from Kevin Flanigan's lab where it works very well for duplication, especially for exon 2 duplication. And so when you have a duplication of exon 2, it puts you out of frame. And then you can put in a U7 snRNA, and of course you're going to have 2 binding sites because it's the exact same genetic material in each one of those sequences. And so it comes up, you'll have this scenario where what is the outcome if you have 2 of these U7 snRNAs binding? And so it's kind of like at the craps table, your chances aren't always 100%, right? It's sometimes one will bind to one of the exons, and then sometimes one will bind to the other exon too. And if that happens, only one of them will be removed, and that's a great outcome because it will throw it back in frame, and you'll have a wild-type transcript giving you wild-type dystrophin protein. If both bind, so both the exons are bound by U7 snRNA, they will both be removed, and now you're out of frame. But nature in molecular genetics is wild, right? And you got to be really observant. If you're observant and you start looking, sometimes you find something that you weren't expected. As it turns out, there's an internal ribosome entry sequence in exon 5 that is revealed by the deletion of these exons that then allows for truncation or the production of an N-terminally truncated dystrophin that has high function. Oh, there we go, back. Okay, and so the outcome may have looked bad, but by close examination, it's actually not a bad outcome. Okay, so delivery. So we heard in the previous talk by Kevin that there's U7 but there's ASO approach, and I've discussed here U7 snRNA. So why would you pick one over the other? And the answer is there's benefits and there's weaknesses to each one of them. So the benefit, I think, for U7 snRNA is that it's long-lasting if delivered by AAV. So this would result in an infrequent, delivery or just a one-time therapeutic. And then the benefit of ASOs for exon skipping is that they do not have an immune response. And so there's no innate immunity to ASOs, the way that they've been modified and all of the work that's been put into them, where there is, as we all know, an innate immunity. Some people have innate immunity to the AAV capsid. So the future of U7 siRNA, and maybe Kevin can talk about this during the question and answer period, is that there's an ongoing clinical trial. And that if successful, the results from this first-in-class trial may provide opportunity for targeting other exons for therapeutics. So really the take home from this is that there are multiple irons in the fire for development of DMD therapeutics. Each approach has strength, each approach has weaknesses, and I always say it's too early to pick a winner, or maybe we shouldn't pick a winner. We should advance many to the clinic to see which one will work best for everybody. And it's good to invest in new ideas to find one that may be a la carte, or maybe one that will be a platform therapeutic for many. And with that, thank you, and I could take questions.
I'm going to invite, all of our speakers to come up and take a seat. And so if you have questions about exon skipping, about stop codon read-through, dystrophin expression, anything else, we have mics in the middle of the aisles. Happy to take those questions now during some of this discussion. I think while we wait for questions, I did want to kind of start with maybe one for all of you. We talked a little bit in the discussion around muscle content adjusted reads of reading out dystrophin expression. And so this is something that we're seeing in some of, a number of presentations. Whenever companies talk about data, there is here's the biopsy and the expression, and here's the muscle content adjusted, or muscle adjusted content. Can we just talk through again what that is for families and maybe why we're using that sometimes?
Sure. Right. So first I'll say there are several different ways to approach this. That's one thing to say. And I would say that I think it's really important for any sponsor doing a trial or anybody publishing a trial to at the very least be transparent and make it perfectly clear in the methods section how you're doing that. That's not always been the case, I would say. So there's two questions, I think, the validity of it and how it's done. Maybe it's fair to say. There are several ways that you can take the area of a the most common way is to look at a cross-sectional area of a biopsy to calculate the degree of that cross-sectional area that consists of contractile elements. We can do this in the same way using the sort of imaging tools that Dr. Nelson showed earlier before. And then take that as a fraction of the cross-sectional area and use that as a correctional way to say, well, what is the dystrophin content only of those areas that are contractile? One can do it several other ways. One can do it with homogenized tissue, looking for non-contractile proteins, collagen or other things. Other people have used staining to look for collagen across the region of cross-sectional area. Each of them are different ways to do it. One question is, how valid are they? They're out there in common use, so as long as people are making clear how they're done, it's fair to make these comparisons across it. We have to recognize that ones that depend on cross-sectional area, when we do a Western blot, we're usually not doing it on the next slice down from a cross-sectional area, so it might be imperfect. In my opinion, it's an imperfect measure. It's in common use. We're obligated to share the methodology about it to make some comparisons about it. So I don't think it's without value, but that's why it's done, to try to accommodate for that. I'm sure Stan probably has other thoughts related to it as well, or maybe not.
Yeah, I mean, I would just reiterate, I think it's conceptually, any time that we're trying to restore dystrophin, at least for the myofibers, you want to be restoring it in the myofibers. So if you have fewer myofibers, you probably can't restore as much dystrophin no matter how good your drug is. So it's probably the fairer way to do it. And I think it's going to become more the norm. And as Kevin said, there's 1,000 different ways to normalize for how much myofiber you have. And I think there's a lot of nuances to that, unfortunately.
All right, we'll start here in the middle.
Hi, I'm Dominic Fullenkamp, Northwestern. I'm an adult cardiologist. We're always looking to see if we can, at every patient we see, to make sure that they're not a candidate for one of the approved exon skipping therapies, and trying to get them approved as adults. But why aren't we studying these patients clinically and using the cardiac outcomes, in exon skipping as the surrogate? And obviously it's next to impossible to do a cardiac biopsy in these patients, but why aren't we using the cardiac outcomes and studying them?
Can I just steer you to the preclinical data from the development programs, particularly I think the TFR1 ones are the ones that are most obvious to me. Where in preclinical studies, one of the advantages of the transferrin receptor approach is it is much higher uptake of the active PMOs in muscle, just from PMO concentration in all the animal studies. Frankly, the naked PMOs don't get into heart very well, so its utility as an outcome measure is probably, they're probably much less protective of heart than we would wish.
And then we'll over here on the
Yeah,
left.
it's a question for John Luke. So your U7 snRNA technique, is it applicable to other deletions? For example, if there is a 16 and 17 deletion, can it be used for skipping 18, for example? How easy it is or how difficult it is?
So that might be a better question for Kevin. So I was tasked for describing how it worked, but I think Kevin's lab is the one that is actively using it.
Right, we are. That's fine. No, we have a lot of cell lines from patients where we've studied these. As you might know, we have a lot of cell lines. So what we do when those of you who are kind enough to give us skin biopsies for research purposes, we take those skin biopsies and we grow fibroblasts out of them, and we put in a gene that turns them into myoblasts, to muscle-like cells. We can never call them true myoblasts, but they behave in every way like myoblasts. We put them in the, and then once we put them into that lineage, then they're expressing dystrophin, and we can do these kind of manipulations to it. So we are very much viewing this as a platform approach, and we have essentially U7 RNA targets for every single exon, and we've studied this repeatedly in many different ones, including 18, including other ones. And we're really now at the point, we've talked about it for years, but we're really at the point where we're going to seek guidance from the FDA about a platform approach to this, with the idea that if all we're really changing is the targeting sequence, what's the minimal amount of safety data we can do to move forward? And we're optimistic about that possibility. At the same time, we're looking, there's some other ways to try to think about multi-exon skipping or multiple different targets on one U7 and so on that might be relevant as well. But it's really applicable to just about every exon, and we have loads of examples of this. In our lab, we're always like, well, let's do one more. Let's do 3 more cell lines before we do it. Let's do one more experiment. I think we're all familiar with that, but I think we're at the point where we're going to draw a line over it and seek guidance from the FDA about the platform approach to it.
Okay, thank you.
Yeah, and I think this is actually a good point, because oftentimes what we hear from families is that with the exon skipping approaches and things like the U7 technology, a lot of these could be applied to other skip amenable mutations, but we do see a lot of it basically concentrated in the hot spot between kind of 45 and 55 of kind of what we're targeting. So could you just, Kevin, talk a little bit more, when you're saying this kind of platform approach in terms of kind of the FDA side, because this is, I think, we're starting to talk about it a little bit more. We've talked with the community about it, but I think it's helpful to kind of drill down a little bit more into this.
Right. Well, so the idea would be to allow approval of a what it exactly means is yet to be seen in guidance with the FDA, but the idea would be to have minimal requirements for repetition of safety studies and efficacy studies once we've established those baselines in individual, particularly after we've done a first-in-human study and can show the safety of it. And not have to repeat the manufacturing approvals for every single one we do when we're doing it exactly the same way. I think I just want to throw in here this, a challenge for this would be taking this approach to make U7s individualized doses for rare mutations. Or we didn't talk about the class of mutations that I know we're interested in, where deep intronic mutations make a portion of the intron be included as what's called a pseudoexon. Those are great candidates for skipping because you make wild-type dystrophin out of them if you can. It's hard to come up with a commercial model to make an individual single-patient dose for those. It's not impossible to think about that. But I want to also mention the companies that have these second-generation exon skipping things. Obviously, they're running trials to get success based on exons, based on how many patients they can treat. But there's no reason if one of these doesn't work exceedingly well that you couldn't use those therapies for individualized exons if we can talk those companies into doing it and maybe come up with a commercial model that would say it's probably cheaper to make a PMO and conjugate it to an antibody than it is to make doses of AAV. Could we come up with a model where we could really do individualized therapies based on those? I remain optimistic about that.
Thank you. Question over here on the right.
So actually, I like Kevin's answer there because my son, he has a splice site mutation in intron 62. So I think pseudo exon skipping would be good for him, potentially full-length dystrophin, if he could get that. So I'm anxious to see more research on that.
I'll say we have done, we published a paper a few years back on a whole bunch of these pseudo exon mutations, and we have cell lines from a variety of patients where we've done this approach, and we can clearly show full-length dystrophin expression with them. So that's one of the keys of our sort of platform thought to it. And if we don't have your son's skin cells, we'd love to talk about getting them sometime.
Thank you.
Anthony in the center.
Hello. So my little guy at 4.5 has 44 to 55 deletion. My wife told me not to believe everything chat tells me, but it says that it presents more as a Becker symptoms, and I'm just wondering if there's any truth to that.
I'm happy to get together with you later and go through some of the databases we have on it. 44 to 55 is one that, there's a lot that I could jump and offer you guidance on. That's not one of them, but we have tools to kind of look at those ones. It's out of frame, I know that, so I would like to sort of see what options there might be for it. If you don't mind, we can get together later on it. I'm sorry I can't recall the details of it.
Thank you.
And then over here.
Does U7 sRNA approach will be useful for the frame shift mutation also?
It can be if the frame shift occurs in an exon that you could splice out and put back into frame. So if the frame shift occurs in exon 44, you could basically put it back into frame by removing that exon. Does that make sense?
Yeah.
Although for many of those might be dual exon skipping strategies. So most exons, except for in the area where you were targeting the nonsense, all the square ones, if there's a frame shift in those, you want to just remove those individually. There's a theoretical possibility if you had a frame shift in exon 50, you'd need to remove 50 and 51, right? So I think the efficiency of the approaches will matter greatly.
Mm-hmm. All right, and I think that was it from the questions from the audience. So I would like to thank our presenters for some great talks this afternoon. And then we will do a quick round of polling before we break, but thank you all.